it's my pleasure to introduce Vivek Hari Sundar. He is currently a postdoctoral fellow in St. Louis at the Donald Danforth Plant Science Center in Keith Slotkin's lab. But today he's actually going to tell us about some of the really fascinating work that he did during his PhD at the National Center for Biological Sciences at the Tata Institute of Fundamental Research in Bangalore, where he's been working on Histone H4 variants as well as RNA polymerase 4 in plants. And I'm really excited to see what Vivek has to show us today. So please, Vivek, take it away.
Thank you. Thank you for your introduction, and thanks to the organizers for the opportunity to share this work. So today I'll be talking about how a Oryzae, a rice family specific Histone H4 variant is important for salt stress responses in plants in general. So chromatin can be broadly classified into euchromatin and heterochromatin, depending on how closely it is packed and how accessible it is to the gene machineries. So broadly, what we are seeing here is a Arabidopsis nucleus stained with DAPI, such as with dark spots indicating the condensed heterochromatin, and counterstained with different antibodies specific to different histone modifications. What you can appreciate is that the euchromatin and heterochromatin are distinctly decorated by these several histone modifications, and they are important regulatory elements in the genome. In plants and in any eukaryotes in general, what is known is several histone modifications, DNA methylations, histone variants, and non-coding RNAs together contribute to regulating genes, leading to specific transcriptional profile. So what I'm showing here is currently a fundamental unit of chromatin called as nucleosome. Nucleosomes are made of 4 types of histones: H2A, H2B, H3, and H4, and they assemble into these beautiful structures called nucleosomes, wrapping the DNA around it. So this is the fundamental unit of packaging the DNA in eukaryotes. And among eukaryotes, what we also see is, in addition to these well-known canonical histones, there are histone variants. So these histone variants are nothing but sequence variants of these proteins, which conserves the property of a histone, but incorporates several modifications or variations that confer specific properties. So these histone variants are distinguished by which kind of chaperones and modifiers they interact with, when they are expressed, is it stimulus dependent, and things like that. So all of these histones, when they form nucleosomes on the chromatin, creates a huge diversity, leading to different kinds of nucleosomes shuffling around in the chromatin. So the outcome of this is incorporation of this variants can have transcriptional regulatory roles. They can either enhance or inhibit transcription by changing the chromatin states, accessibility route to transcription factors, polymerases, etc. So how these histone variants dictate these properties is because of its specific variations. So here, let's say this is a typical canonical nucleosome. Incorporation of histone variants lead to either variation in how it is packing the DNA, how it is wrapping the DNA around itself, whether it is leading to a flexible DNA ends, or it is keeping it compact, or how stable it is to be acted upon by, let's say, chromatin remodelers and things like that. So I'm showing here an example of a histone variant called SENPA. When it is incorporated, what we see is it leads to flexible DNA ends. This property allows it to have open nucleosome structure. So these kind of variations contribute to specific molecular properties that can dictate regulatory roles. So in plants, there are several histone variants that are recognized, most of which are conserved among all eukaryotes, for example, SNH3 and H2AZ, H2AX, where they perform almost conserved roles. But plants do have specific variants like H2AW that are necessary for heterochromatin silencing, transposon regulation, things like that. Uh, so these histone variants are very crucial for plant development and phase transitions. And when you mutate the histone variant or its chaperones, you see drastic phenotypes. And most of the histone variants are essential genes in plants, and you cannot knock them off completely. That leads to embryonic lethality. So what is well known is in a nucleosome, we have 4 histones, and among eukaryotes, there are several variants of H2A, H2B, and H3 have been recognized and found. But variants of H4 is quite rare, and it is seen only in kinetoplastids and nematodes and things like that. But none of the variants of histone H4 have been recognized in plants. So we performed a kind of homolog search of histone H4 among plants, and what we identified is a histone H4 variant, the sequence of which is shown here, and the canonical H4 sequence is shown at the top. So what you can appreciate is most of the histone defining elements and the helices are conserved in the variant, but there is a huge deal of variations in the N-terminal tail. Also, the specific sites which undergo well-known post-translational modifications are absent in the histone H4 variant. So this histone H4 variant, as you could appreciate, is a specific case only in rice family. It is not seen in any other genera in among plants, and what you could see is it is around 73% identical to the canonical histone, and most of the variations are concentrated in the N-terminal tail. This histone is quite well expressed in the rice. These are different tissues of rice, and we see clear expression in several tissues. So to test if this histone variant is indeed incorporated into the chromatin, into the nucleosomes, we generated specific antibodies that recognize only the variant but not the canonical H4, which clearly shows it is incorporated into the chromatin. And also, it is seen in distant relatives of modern rice that we usually use. So it is conserved in distant relatives of rice as well. Uh, the hallmark of any histone is that it forms nucleosomes. So we purified all the histones from rice and in a recombinant fashion and tried to reconstitute it into a nucleosome, and we could clearly see that the variant forms nucleosome, which confirms that it is an authentic histone variant. We also performed ChIP-seq with the specific antibody, and we could see that it occupies generally a heterochromatic region in the genome, which is enriched with H3K9 marks. So we had a fantastic collaboration with JP's lab, and we tried to solve the structure of canonical histone H4 containing nucleosome and the variant histone H4 containing nucleosome. What came out as a big surprise is like, unlike metazoa and nucleosomes, the DNA is not very well packed in plant nucleosomes. This is the overlay of both the structures. What we could see is that DNA is not well packed in the canonical nucleosome, while in the variant it is condensed. So basically, the nucleosomes in plants in general are accessible. They do not wrap the DNA well, while presence of variant makes it condensed. So this was really a big surprise because in metazoans, it is the DNA is usually well wrapped with a proper canonical histone H4. So to test this, we did a MNase titration. So what we do is we take these nucleosomes and do a time course titration with micrococcal nuclease, which chops off the free ends of the DNA, and do a deep sequencing to understand which regions of the nucleosome core particle are protected. So when we did that, we could clearly see that compared to canonical nucleosome, which is more accessible, leading to shorter DNA fragments, presence of a histone H4 variant protected the nucleosomes, protected the DNA better, leading to protection of the DNA ends. We also generated a mutant. This is a structure-guided mutagenesis where we recognize that this particular residue is responsible for close packing of the nucleosome. So we mutated this residue, and clearly it reverts back to open state. So suggesting presence of this histone H4 variant makes nucleosomes more condensed. So another important aspect of histone variants is to change the stability of the nucleosome. So this is a very important parameter in terms of regulatory role, because how easy a nucleosome can be open directly correlates with how well it can be acted upon by chromatin remodelers, modifiers, and other gene regulatory machineries. So to assess this, we use nucleosomes and a fluorescent reporting dye called a cypro-orange, which fluoresces only with the denatured protein peptides. So nucleosomes decay in two steps. So first, it eliminate upon heating or presence of a chiotrope, it decays in two steps. First is the elimination of H2A, H2B, and then is the elimination of H3H4, leading to complete dissociation. So it's a bimodal process, which we can trace using the fluorescence from the cypro dye. So here, what you see here is a decay profile, thermal decay profile of nucleosomes of canonical and the variant histone-containing type. And clearly, the first peak is the H2A, H2B decay, which happens uniformly in all the cases. But the second peak, which corresponds with H3, H4 decay, shows difference at which the decay occurs. The canonical, which is shown here, is quite stable, while presence of variant leads to the instability. We also use the structure-guided mutant, which is V33A, that shows the intermediate profile, suggesting that the instability in the H4 variant is due to contribution from the DNA packing, which is mediated through this particular residue. So H4 variant is leading to less stable but condensed nucleosomes. So how it affects the plants? So we generated a CRISPR-Cas9 mediated knockout of this particular variant in rice. What we could readily observe is that these plants show a stunted growth. Also, they make smaller grains, and when you overexpress it, it leads to sterility, and there are whole sort of other phenotypes associated with meddling with this particular histone H4 variant. So when we did a transcriptome on this, what readily stood out is several of the stress responsive transcription factors are misregulated in the knockout, which you could see here. So to clearly point out which kind of stress response it is associated with, we used a publicly available data set of several stress phenotypes, which people have already done. And when we compared the gene misregulation of the H4 variant knockout with several other metallic and abiotic stresses, what we find is it closely resembles that of a salt stress state. So this is even true when we did a reciprocal pool of genes where we picked the differentially expressed genes on salt stress, but still the correlation held true. So this led us to believe that H4 variant knockout is somehow creating a precocious salt stress state. To test this further, we performed salt stress in the plants, and what we could clearly see is the salt stress response closely correlates with the knockout knockout response, which is seen in this scatter plot. So this is quite surprising. So to test what happens to H4 variant upon salt stress, we performed immunofluorescence microscopy with a control and with a H4 variant specific antibody, and we could clearly see that the occupancy of the variant is more upon salt stress. The ChIP-seq also shows that compared to the no stress state, where it is mostly in the heterochromatin region, upon salt stress, it occupies a wider gene-rich regions. So this was to understand how this H4 variant actually controls the salt stress responses. We checked the level of another histone mark, H4K5 acetylation. Just to recall, the histone H4 variant itself cannot undergo the acetylation mark because this histone variant lacks that residue. So what we find is in the knockout, the K5 acetylation marks at the H4 variant peaks are almost abolished, and the protein coding genes that overlap with the newly occupied salt stress specific H4 variant is necessary for K5 acetylation spike that usually happens in wild type, which is what is shown here. So how it changes the global profile of K5 acetylation mark is a long-standing question. To test that, we perform ChIP-seq with and without salt stress in wild type and knockout. And what came out as a surprise is generally H4 variant helps in demarcating where the K5 acetylation should happen. And when we knock it out, the K5 acetylation marks are lost in several thousands of loci, which you could see here. But surprisingly, these are the same set of loci which lose the H4K5 marks upon salt stress. So this kind of resemblance in both transcriptome and K5 acetyl profiles led us to believe that H4 variant is responsible for controlling salt responsive K5 acetylation deposition. And in the knockouts, this is completely precocious in nature, and the acetylation happens in a precocious manner. So in conclusion, what you could see is in the normal state without any stress, the H4 variant nucleosomes are mostly concentrated in the heterochromatin. But upon salt stress, there is an expansion in H4 variant occupancy leading to euchromatic or protein coding gene rich region incorporation of this variant, and presence of this is responsible or kind of contributes to how easy the nucleosomes are modified by K5 acetylation marks in the neighborhood, leading to a stress response that is seen when we give a salt stress state. So this H4 variant actually acts as a switch for K5 acetylation, leading to how and when the stress transcriptome should be activated. So with this, I would like to thank P.B. Shivaprasad and his lab for fantastic guidance, and JP Lab for the cryo-EM structures, and this was supported by Newton Baba Fund. Any comments and questions are welcome, and this is available as a preprint, which you could access through this. Thank you.
That was great, Vivek. Thanks so much. I guess we can start with the audience. We've got Nicholas has his hand raised. You can unmute yourself now if you'd like to ask the question, Nicholas.
Yes. Hello. That was a very nice talk and a lot of work.
Thank you.
I guess I had a couple questions. So first, I was wondering with your cryo-EM structure, are you using 601 DNA there? And
yes, it's 601 147 base pair DNA. Yeah.
And that's without H1, correct?
Sorry, this is without H1.
That's without H1. Okay, so I guess I was just I also am curious, and I'm just curious how it influenced the structure, but I have my ideas. But I'm also curious about the salt concentration that you use in your structure, and the salt concentrations you use to induce salt stress.
Oh, so this
How those
was.
would affect your temperature experiments.
Oh, okay. So the temperature stability experiments we did at I think 25 millimolar salt, which was kept stable throughout. But we have tested the salt sensitivity of the nucleosome with like both canonical and the variant containing nucleosome decay at the same rate up with different salt concentration. It's not that it is only the canonical that decays faster. So yeah, that's not shown here, but we had tested it. It's not sensitive to the salt directly.
Thank you very much.
Yeah.
Great. So in the interest of time, I'm going to ask Jihu, who has their hand raised, to please enter your question in the Q&A so that Vivek can answer it there. And it looks like there's already another question in the Q&A. So
Yeah,
Vivek,
thank you.
thanks again.